discovered a new protein called Importin Alpha 16, because of which my papers were very highly cited, because nobody had heard or discovered this protein in the past. So think in a very technology agnostic way. What is the best way to get to the solution? And if it is a skill you don't have in your toolkit, get it. Or bring a collaborator in, or bring a partner in. If you have a hammer, every problem is a nail, right? But what I'm asking you to do is don't be kind of bound by that constraint. There are many, many other technologies you can use as well to find the right solution. And I thought that is a very, very interesting problem to be working on, because the day I turned 45, my physician told me that you need to get a colonoscopy done to see whether you're at risk for colon cancer. And I do not want to undergo a colonoscopy. I didn't want to spend one full day with that. I asked if there's a blood test, and there is no blood test. So when I found out about Freenome that they're working on developing a blood test for colon cancer risk, I thought, what a great idea. Like, instead of going through colonoscopy, if I can just give a blood sample and figure out, am I at risk for colon cancer? That's a great product to have. I actually had ambitions to be in medical school. I would have liked to be a doctor as well. But I realized that what was attracting me more was the biology. All the action is happening at the cellular level. It's happening at the molecular level. Things are going wrong in your cells because of which you're developing disease. And I thought that was so much more fascinating to understand. Think about how quickly we can start diagnosing cancer, even before you have the symptoms. Think about what that will do to the future of healthcare. That is what is now in sight if we make the right moves over the next 10 to 15 years.
On today's episode of the Inference Show, we are joined by Dr. Suraj Saksena. Dr. Saksena is a clinical chemist whose career has spanned drug discovery, biotech innovation, and clinical diagnostics. Suraj is currently the Vice President of Product Development at Universal Diagnostics. Suraj has led scientific and operational teams at Freenome, Adaptive Biotechnologies, BD, and Theranos. Suraj has a PhD in biochemistry from Texas A&M University and an executive MBA from Quantic. In this episode of the Inference Show, we will explore how Suraj is shaping the future of the diagnostics industry. Suraj, welcome to the Inference Show.
Thanks for having me, Manish. Great to be here.
Awesome. Suraj, your career bridges research, product development, and leadership. Where did it all start?
Wow. Started way back in India. After I was done with high school, I pursued a bachelor's degree in biochemistry with a real keen interest in trying to understand, human health and disease at the molecular level. So anytime you develop a disease, what's happening, what's going wrong in the cells, what molecules are out of whack, why are we getting that disease, how do we diagnose those diseases, how do we find therapeutics or cure for those diseases. That was a very keen interest I had, and that led me to pursue a bachelor's degree in biochemistry. So I went to the University of Delhi for that, then pursued a master's in biochemistry as well from the University of Delhi. And then in the year 2000, about 25 years ago, moved to the US to pursue a PhD in biochemistry as well. I was very keen for my graduate work or my PhD work to work on a very specific problem known as membrane protein trafficking, which is a cell biology problem about how do proteins get to certain different membranes within the cell. And I chose to work with a PI based in Texas A&M. So I came to Texas A&M for my graduate work.
It seemed very early on, you kind of knew what you wanted to do.
Yeah, I think even sort of very early in life, growing as a child, I was always a very inquisitive, very curious child. And I think when you have some of those traits of being very curious, asking a lot of questions, those kind of traits typically propel you towards a career that's more focused around the lab or being around the bench and really being in a field which is more anchored around research and discovery and inventions. So that really kind of propelled me in that direction. And then maybe on a more personal level, I lost my father at a very young age to a cardiac arrest or cardiovascular disease. And that really shaped me in many ways, Manish. It really made me very motivated to pursue a career in life sciences and really understand health and disease. And with that sort of impetus behind me, I think that's brought me to where I am today.
And how old were you when he passed away?
I was 9. So in the very formative years of my life and my journey, I lost my father. And then that, like I said, really opened my eyes towards how do I make my life, my career, something that will move the needle in terms of understanding disease and finding cures or finding diagnostics for them.
Were there signs, or was it a very sudden thing that happened?
Very sudden. It was a cardiac arrest. We didn't have any forewarning. There were no symptoms. My dad was a very healthy man, but the disease was slowly progressing. Now we're talking about back in the 80s when there weren't really diagnostics to figure out how are you doing on an annual basis. There wasn't a concept of frequent checkups or whatever. So it was a complete surprise, not just for me, for our entire family, that to see a very young man, 40 years old, just pass away was a huge jolt. And in so many ways, I think that has shaped the man I've become today and the career that I have today.
Understood, Suraj. And Suraj, how was life in Delhi in the 1980s, 1990s?
Life was very different. I mean, every year that I've gone to India since I moved to the US has been, India keeps changing very, very frequently, very rapidly. But back then, life was a lot more laid back. There was no internet, there was no social media, there was no distractions. As a student, it was all about, you go to school, you come back, you do your homework, and then you play, and then you just focus on your studies. You have exams coming up, you have assignments that are due. So it was a very simple life, more sort of anchored around the family, career, and so on. I would also say again that, and not to belabor this point too much, but I think once you lose a father at a young age, typically you go from a boy to a man very quickly because you realize the responsibilities that you have on your shoulders. So I think I lost out on a lot of things where I couldn't be the guy who was just playing around or bunking classes or going to watch movies or whatever. I had to be very serious, very focused on my career because I knew my mother was counting on my success.
Suraj, I lost my father a few years ago, and right after I felt that today is the day I've become a man from a boy.
You mature very, very quickly. I think it's very safe to say that.
Yeah, but you had to go through that very early on. Out of that experience came your desire to understand from what, because it was so sudden. Because you could have chosen to be a doctor as well, right? Why did you take this path?
I actually had ambitions to be in medical school. I would have liked to be a doctor as well. But at some point in my career, like when I was even in high school, I realized that what was attracting me more was the biology. It was less around the medical science, but more around what goes wrong in cells. What happens, if you try to understand heart disease, or if you want to understand diabetes, or you want to understand cancer, all the action is happening at the cellular level. It's happening at the molecular level. Things are going wrong in your cells because of which you're developing disease. And I thought that was so much more fascinating to understand. Because if you're just a doctor, you look at symptoms and then you prescribe a therapy or you prescribe therapeutics. But to be a scientist who's understanding the disease at that foundational level, at the molecular level, I just found that incredibly fascinating. And that's why I then pursued a direction which was more anchored towards biochemistry and really understanding what goes wrong in disease.
Understood. And how was studying at University of Delhi?
It was great. I was at the Department of Biochemistry. It was a very elite department, if I can say that. We had only 15 people when I was in the bachelor's program because they would only select the cream of the cream. But a very good department. It provided me some very, very good foundations in understanding cell biology, molecular biology, genetics. And even when I came to the US, I thought I did very well in school here because my foundation from India was very strong. I think the bachelor's, the master's degree I got over there really prepared me for graduate work and then postgraduate work here in the US.
And then right after your master's, you came to Texas A&M College Station. How was College Station?
It was a huge cultural shock coming from Delhi, which is one of the biggest cities in the world, coming to a very, very small college town outside Austin. It's about 100 miles from Austin. And the only thing that's there in the town is the university. Everything in that town revolves around the school. So all the employment in there, all the jobs that are there, all the shops that are around are all related to the school. Very, very different experience. So it was not just a cultural shock of coming to a different country, but coming to a different country and living in a very, very small place. So that took a lot of time to kind of adjust to. But I would say that it also motivated me to spend a lot more time with my books and really be in the library and just keep studying for my courses. Overall, by the time I left College Station 5 years later after I got my PhD, I really missed it because it was small, laid back, very low traffic. It was very, very easy to live in and very, very low cost of living as well.
Yeah, you could probably get you could get an apartment in Austin for like $500, like a one bedroom. I don't know
easily
what you could do in College Station.
My very first apartment was $400 that I shared with somebody, so I paid $200. That was my cost of living, was $200 on an apartment.
Yeah, awesome. And then during that time, From what I understand, you wrote some pretty awesome papers, very well cited. What was your research topic and whom did you work with?
Yeah, so I think, like I said, I came to Texas A&M because I really wanted to work with this particular faculty member. My graduate work was focused on a very basic question of cell biology, which is how do proteins get to the nuclear membrane? So the nucleus is an organelle within a cell, which is the basic unit of life. And a very interesting question is how do the proteins get to that location? Now that's a very important question because if certain nuclear proteins don't get to the membranes correctly, or if they're not at their correct location, you can develop some very debilitating diseases. So there's a bunch of nuclear proteins called lamins. And if those lamins don't get to the nuclear membrane, you can basically develop a whole range of diseases known as laminopathies, where you have a lot of muscle disease, you have muscle weakness, you can have cardiac problems, you can have premature aging. So all the work I was doing had a very tangible impact on in the real world because this had a direct connection with disease. Over the course of my 5 years of graduate work, what I did was I was understanding that mechanism at a molecular level. How do proteins get there? Are there other proteins that are helping them get there? Once they get there, how are they anchored in the membranes? So very, very basic sort of textbook questions for cell biology, but using some very sophisticated techniques of biophysics like fluorescence spectroscopy or photocross-linking. So it was a very, very interesting project. And over the course of these 5 years, I actually discovered a very novel protein. I discovered a new protein called Importin Alpha 16, because of which my papers were very highly cited because nobody had heard or discovered this protein in the past. And I showed that if you don't have this protein in the cell, then your nuclear proteins will not get to their correct location. Then you will develop those laminopathies that I described. So it became a very tangible project. I discovered something new, and that resulted in some very, very highly cited publications.
Why is it that in your field they don't name the protein after the person who invented it?
Well,
who discovered it? Apologize.
We called it Importin Alpha 16 because it came from that family of proteins called Importin Alpha. It was a smaller isoform of that. But yeah, importins are very, very well known, highly characterized proteins. I just found a new or discovered a new isoform of it. So we called it Importin Alpha 16.
I thought it would be like comets, but no. Awesome, Suraj. And then, right after, I think you were very academic in nature. Did you consider staying in academia?
Very much so. There was, in fact, after my postdoc, so I did a postdoc with Scott Emmer. I was initially at UCSD and then moved to Cornell with him. And did my postdoc for about 3 years. I published 10 papers in 3 years. And I covered all the journals like Cell, Nature, and everything. So there was a lot of interest in me staying in academia. In fact, the first time I told my postdoc advisor that I want to join the industry, he was like, no, you are meant to be in academia. But I was very clear, Manish, in my mind that I wanted to be more in the industry. I wanted to be in the biotech side. I wanted to be more sort of on the applied side of science, less around basic science, fundamental science, and less focused on writing grants or publishing papers. I wanted to be closer to the bedside, having more direct impact on patients and healthcare and so on. So that switch was very natural for me.
Understood. So you started your postdoc at in San Diego, and then went diagonally to the other side of the country to Ithaca. How were those two places in the early 2000s, and how was that transition?
Everyone who heard about that move of mine said, you are moving in the wrong direction. You were supposed to move from Ithaca to San Diego, not vice versa. Look, I took it because, one, I thought Cornell is a great school. Not that UCSD was any less. UCSD is a fantastic school. But Cornell was, again, a fantastic school. The other thing was my postdoc advisor was becoming the dean of the department. And I thought it would have been a great opportunity for me to be there, help him build that department and see how he sets up the entire department, the program, and so on. And then lastly, I liked my postdoc advisor a lot. I didn't want to leave him midway, and so I went with him. And in terms of the places, again, Ithaca was sort of reminding me of College Station. Again, a very small college town in upstate New York, very cold. Almost 6 months of the year, you're sort of covered in snow. And somebody who grew up in India, that was very new for me, and it took a lot of getting used to. But I'll say I stayed there for about a year and a half. I finished my postdoc, and I came straight back to California.
Understood. And then you joined Amicus Therapeutics,
yes.
Right? That was the first company. Where was that, and what did you do?
So Amicus was a very interesting company. It was headquartered in New Jersey, but they were starting a small program on neurodegeneration. And they were really focused on looking at can we solve Alzheimer's disease, which is a growing problem in the US? Can we solve that by developing therapeutics that target a particular cellular mechanism that is known as lysosomal waste removal? So in your cells, you have these organelles called lysosomes that are responsible for removing all of your cellular waste. Now, if those lysosomes don't work properly, you will start accumulating a lot of waste. And typically what happens in Alzheimer's is that your cells, your neurons, are basically unable to turn over all the waste that is being generated in this cell. So it was a very, very unique idea that if we can target the lysosomes inside the neurons, can we basically get some relief from the symptoms of Alzheimer's disease? I thought that was a very, very interesting concept. So I joined Amicus. The neurodegeneration program was based in San Diego. So I went to San Diego and worked with Amicus on their neurodegeneration problem.
I mean, I recently read somewhere that sleep is very important because it removes the waste, right?
Yes.
Which, that's why brain
Absolutely.
health and Alzheimer's, right? Sleep is a lack of sleep increases your risk of Alzheimer's dramatically, right? Suraj, how much do we know of the cells right now? It seems that you've always been reading about it and you've done some fundamental work there. You've been working and studying them since the 90s now. But in your opinion, do we know 10%, 20, 90? How much of the cells do we understand now?
I would say we know a lot. We understand the genome pretty well now because we can sequence the genome very easily. We have a very good understanding of the proteome. We can figure out all the different proteins that you have. We have a very good understanding of protein structure, protein function, protein trafficking. I think our knowledge is increasing day by day. What is still missing and what still needs to be worked on is sort of kind of connecting the dots. So when you have a particular disease, what is going wrong in which protein and how can that be corrected? I think those are things we're still working out. But as it relates to kind of understanding the big molecules of life, the DNA, the proteins and so on, I think we have a very good handle on that with the technologies we have today.
And then the work that you did at Amicus, how far along did it go in the journey of getting out to people, or was it early stage and exploratory?
It was very, very early stage. It was all exploratory, but I really enjoyed it because it was my first industry job. So I did all my sort of cultural shift from academia to industry at Amicus. It was my first foray in the therapeutics world. And so it gave me a very, very good foundation or understanding of drug discovery, drug development, the whole clinical trials that have to be part of drug development and so on. But I did all the work which was preclinical, very, very early stage, working with small animal models, didn't really get to any human trials, but a very, very good understanding of the drug discovery process.
And what was the key difference going from academia to industry?
Oh, huge. There's such a huge sort of cultural shift that happens when you make the switch from academia to industry, right? Typically, when you think about academia, you're thinking about sort of research, which is a bit more open-ended, it's more exploratory, much more slower. In industry, it's very, very structured. You have a lot more processes. Everything is anchored around timelines and deliverables. If you think about academia, it's a bit more, all of your sort of focus is on writing grants, publishing papers, textbook chapters, and so on. In industry, it's all about product launches. How quickly can you launch this product? How quickly can you get it FDA approved and so on? So it's a very, very different mindset. Academia, again, is you can be very kind of single-minded, solo-focused in that. In industry, you have to work very cross-functionally to develop a product or to develop something that can be launched. So it's a very, very different mindset that you have to kind of inculcate.
I mean, you seem to have enjoyed that transition, right?
Very much so. And I think there were several learnings over there. I think when I moved into industry, the first thing that I figured out was I have to very quickly understand the language of business because I didn't have that background. And I think one thing that I did early in my career was I found a mentor who could really teach me or help me understand the language of business. What do I mean by that? Simple concepts of like OPEX, CAPEX, how to write a budget, how do you basically come up with a full budget for a program or a project? How do you look at a business case? How do you write a business case? How do you evaluate the commercial opportunity for a given product or whatever? I think those are very, very basic concepts that I learned very early on from a mentor, and those were very, very useful. I also took some very initial courses, sort of very entry-level courses in project management, corporate project management. How do you build a roadmap? What are phase exits? What are Gantt? How do you come up with resource allocation? I think those early learnings have really, really made me a lot more seasoned in my career.
And then 2011 or so, you started working at Theranos, which was the period of the rise of that
Yes,
company, right?
yes. It was the golden days of the company. I came in and I was asked to basically develop a program for reagent development. Basically, when you run a diagnostic test, you require a bunch of reagents that have to be mixed with your sample, your blood sample or your stool sample or whatever sample you may have given. And those reagents have to be developed, right? Meaning you have to produce them, you have to qualify them and so on. And I basically joined the company and was asked to build a team that would develop all those reagents, QC all those reagents, and basically release them for diagnostic testing. Started as a pretty early employee in the company, built a very large team that was focused on reagent development and got a lot of experience in kind of clinical reagent development over there.
Yeah, you were the 50th employee or so?
Yeah, I was pretty early in the journey there.
And then 2015, of course, things happened. But then you moved on to Becton Dickinson, right?
Yes.
Which is a global giant, right?
Absolutely.
A very, very large company.
Very large company. Yep. They have many, many different products, from instruments for flow cytometry to reagents for flow cytometry to diagnostics. It's a global giant.
You stayed there for about 5 or 6 years?
5 years, yeah.
What kind of work did you do there?
Many different roles over there. I was sort of leading the clinical reagent development first, clinical assay development. And towards the later part of my tenure there, I sort of was overseeing all custom reagent development. So we were basically developing products that were sort of off catalog, but made sort of in a custom way for different customers and so on. So I wore many, many different hats at BD for 5 years that I was there.
What was the role like? Was it more technology centric? Was it leading a team? Was it
all of the above?
All of the above.
All of the above. So it was a very clear responsibilities around sort of reagent development, assay development, leading a pretty large team that was working on asset development, overseeing the operations group, overseeing some marketing and sales activities, working very closely with med affairs and scientific affairs. So it sort of covered all those aspects that you outlined.
And then 2021, 22, you worked at Adaptive
Yes.
Biotechnologies. What kind of work did you do there?
So it was my first foray into NGS, or next generation sequencing. I really wanted to get into the field of NGS because I think that the future of diagnostics will be NGS, where given how cheap DNA sequencing is becoming, NGS will really drive all the molecular diagnostics moving forward. So I wanted to get a taste for NGS diagnostics, and so I went to Adaptive Biotechnologies. They've been the pioneers in this space, working on NGS-based diagnostics for leukemias, for lymphomas. And so it gave me a very, very good understanding of how NGS is being used in the clinical lab for sort of cancer detection or cancer diagnosis.
How does the core NGS technology work, Suraj?
It's a fairly well-established technology. It's a very complicated workflow, but fairly well-developed, very commoditized. You can buy kits from many, many different vendors to establish it, but it requires a lot of automation. It requires a lot of deep understanding of how the assay is being run. It is a very complicated workflow. It's a very long workflow, but I would say very, very well commoditized today.
And then 22 to 25, you worked at Freenome,
Yep.
Right? You transitioned into, back to blood testing in some ways, right?
Yeah, so I found the Freenome problem to be very, very interesting. Freenome was again using NGS, but the focus area for the company was more early detection of colon cancer. And I thought that is a very, very interesting problem to be working on, because the day I turned 45, my physician told me that you need to get a colonoscopy done to see whether you're at risk for colon cancer. And I do not want to undergo a colonoscopy. I didn't want to spend one full day with that. I asked if there's a blood test, and there is no blood test. So when I found out about Freenome that they're working on developing a blood test for colon cancer risk, I thought, what a great idea. Like, instead of going through colonoscopy, if I can just give a blood sample and figure out, am I at risk for colon cancer? That's a great product to have. So I joined the company in 2022. I was with them for about 3 years, and the focus really there was, how can we develop a blood test for looking at your risk for colon cancer?
How far is it in development now?
It came out very far. By the time I left, we had finished most of our development studies. I know since my departure, the company has been submitting most of their studies to the FDA, so they're expecting FDA approval fairly quickly.
Awesome. So it's going to be a substitute, to colonoscopies and poop tests as well?
Exactly.
Right.
So right now the options are, you can either go get a colonoscopy done, which, like I said, it's a one full day. There's a lot of bowel prep you have to do. It's painful in some cases. It's not the most pleasant experience. The other option you have is to poop in a can, which also is not a very pleasant experience either. So to basically come up with a product which bypasses a colonoscopy and a poop test and can be a simple blood test is a huge market. And so there are many, many companies that are operating in that space. There's Guardant Health, there's Freenome that I worked at, and now the company that I'm working with now, Universal Diagnostics as well. We are trying to develop a blood-based test for colon cancer.
Yeah, it seems the risk is very, very high, right, for people above 45?
Very much so. Originally, if you looked at the screening guidelines from the USPSTF, originally the guideline was that if you went over 50 years old, then you're at sort of average risk for developing colon cancer. But over the last few years, they have had to drop that from 50 to 45 because there's a huge surge in the number of younger people who are developing colon cancer. This is tied to lifestyle changes today, the dietary habits that people have. We are finding that colon cancer, one, is happening a lot more, it's more prevalent. And the other more worrying thing is it's happening in younger people. It's no longer a problem of older age, it's happening in much, much younger people.
Understood. And then very recently you joined Universal Diagnostics, again working on early cancer detection, blood work. How different does that work from the work at Freenome?
Very similar, I would say, technology-wise. It's also an NGS-based test. Of course, there are differences here and there, but overall, the platform, the approach is very similar. Looking at your genome, looking at your epigenome, and then figuring out, are you at risk for developing colon cancer or not?
So is it more of a Signal C, their core product? Is it a risk assessment or is it a test?
It's a test that will basically tell you whether you have colon cancer or you don't have colon cancer. So we are looking at specific sort of markers on your DNA, and if we find those markers, we'll basically say, well, you are cancer positive, and if we don't, we'll just say you're negative for colon cancer.
And what is your exact role, and what do you do on a day-to-day basis there?
Yeah, so right now we are still in the development phase, meaning that we are developing the different aspects of the product. It's a very complex product. It involves assay development, it involves automation, it involves reagent development, it involves software development. So all these aspects have to kind of come together and work in sync to basically have this test be out there. So I'm overseeing all of those aspects, from assay development to automation development to software development to reagent development, and then working cross-functionally with quality, with regulatory affairs, with clinical operations. All of us are working in unison to basically deliver this product for the market.
Are you close to submitting for approval, or what stage are you guys?
We are in the development phase right now, and our plan is to start some of our validation studies next year, and then go down the path of FDA approval.
Suraj, if you look at your career at a high level, you started with deep life sciences R&D, spent a lot of time in therapeutics, then diagnostics, and then product development leadership now, right? That evolution, was it planned? Did it just so happen? And what was that journey like if you look back at the last 25 years or so?
Yeah, I think I've already talked about sort of what got me interested in life sciences and health and disease and all of that. But I was also very clear, Manish, that I wanted to expose myself to kind of both sides of medicine. If you think about medicine, there are two aspects to it. There are two sides of the coin. One is diagnostics. It's basically figuring out what is the disease that you have. And the other is therapeutics. How do I treat that disease? How do I control the symptoms of the disease? I basically wanted to get an experience in both of them. So I started my career in therapeutics, really understanding drug discovery and drug development. And then I wanted to move over into diagnostics. So then I joined diagnostic companies where I've really spent the last majority of my career working on diagnostics. You can say I enjoyed diagnostics more because I've stayed in it for much longer. But I really think my career kind of went in that progression because I wanted to experience both sides of medicine.
Understood. Suraj, in all of these diverse experiences, there are some key insights that you have developed on various aspects. So let's try to delve deeper into the key learnings that you have had. Suraj, what is your mentoring philosophy when it comes to people? Because people will, nobody could be perfect in everything that they do, right? But, jobs of today demand so many different skills, right? So how do you mentor people into becoming a better version of themselves?
Yeah, I think mentoring is, you always have to be very clear about, it's a very honest conversation that you need to have with somebody. That, look, these are your strengths. I know you're very good in these areas. These are some areas you need to grow and develop. So it's a very honest conversation about, you're here today. how do I get you from here to here? Or you are at this role today, you aspire to be at this role tomorrow. Here's the delta. And how do I help you bridge that delta? So it's a very open, honest conversation, and then basically offering yourself as a bridge to bridge that delta between where they are today and where they want to be. And then once you identify the delta, you are fully committed to helping that person. It cannot be something that you just had a conversation. There has to be complete buy-in from your side that you will do everything that you can to get that person to the next level. And so whether that means I need to send them to take a course somewhere, or I connect them with somebody else who can give them that skill set, or I'm giving them a new experience that they would like to have, you have to be fully committed to helping those people.
Every person that you choose to then mentor, you have to essentially maintain a record of their progress, right? You do that?
You do. It is not easy. And in fact, beyond everything that you've outlined about my career, till date, the thing that I'm most proud of is the people who have mentored or people who worked for me in the past, to see them where they are today, where they've risen in their careers. That is more satisfying than any of the things that you've covered in my introduction here today. I've seen people who were scientists 10 years ago who are directors today. I've seen people who are RAs, research associates 10 years ago, who are associate directors today. And that gives me enormous pride and enormous joy.
Awesome, Suraj. That's something, to look back at. Not only were you successful yourself, but you had some role in the success of others as well. So that's awesome.
One thing you had asked me about my mentor, right?
Yeah.
One thing my mentor told me very early in life, which I've always remembered, always give back. So if somebody is helping you today, help someone tomorrow. And that notion of giving back stayed with me. And in whatever capacity I've done in my career, I've always tried to help people wherever I could. If someone came to me and said, Suraj, I really need this help in my career, if there's anything I could have done, I have tried to do my best.
Suraj, you've spent the last 2 decades or so in working to solve complex scientific problems. What are good approaches to doing that? What works? What doesn't work?
Yeah, that's a very good question, Manish. Let me maybe double click on my graduate work. I think that because that will be a very instructive example. I told you that in my graduate work, I discovered this new protein that helps proteins get to the nuclear membrane, and we talked about that whole story. Just to give you some context around that project was a very basic, fundamental cell biology question. Like I said, how do proteins get to the membrane of the nucleus? To answer that question, I actually use a virology model system. And to answer that question, I also use some of the most sophisticated biophysical techniques that were out there, like photocross-linking or fluorescence spectroscopy and so on. And so what I learned very early in my career is always be very, very solutions focused, but very technology agnostic. So whatever technology will help me find the right solution, I will go for it. And it can be a technology I don't know, and if I don't know, I'll go learn it, or I'll bring in the right collaborator to help me with that. But don't have these blinders on that I know this technology very well, so this will help me find the solution, because you may be barking up the wrong tree. So think in a very technology agnostic way. What is the best way to get to the solution? And if it is a skill you don't have in your toolkit, get it, or bring a collaborator in, or bring a partner in. But don't think that everything I have in my toolkit is all I have. I think that's something I learned very, very early in my career, and it's helped me throughout the course of my career. I can give you many, many examples of that.
So being technology agnostic really helps in solving complex problems.
Absolutely. Most people think about technology, the one that they're the most proficient in. I know this very well. I'm very comfortable with this. So every time I look at a problem, I'm only thinking about this technology, because if you have a hammer, every problem is a nail, right? But what I'm asking you to do is don't be kind of bound by that constraint. There are many, many other technologies you can use as well to find the right solution.
Understood. Suraj, in any interview, you have like 30 minutes to an hour to judge a person, right? What are you looking for?
I think there are 2 or 3 key things that I look for. One is, a good understanding of all that's on their resume. How much did you really deliver? How much were you totally an integral contributor to? I can write 50 things on my resume because I may have very peripherally or very tangentially contributed to them. What I'm looking for is what are the things that you actually drove from start to finish. That's one question that I'm really kind of probing is what are the key deliverables that you personally delivered for a given project or a given program. The other one thing that I put a lot of emphasis on is just drive and motivation. I think that there is no substitute for people who are very energetic and just driven. They want to get stuff done. I think that that's something that I really do look out for. And then people with the right attitude. There are people who can work in very difficult situations, very difficult scenarios because they have the right attitude. And there are people who can have the wrong attitude and just can't get anything done. So I think it's a good mixture of sort of attitude, drive and ambition, and really a solid story of contributions. What have you delivered in different programs or in different companies that you've worked at?
There seems to be a recurring theme, hunger.
Hunger, yeah, absolutely.
There's all this complex technology, there's speed, there's compliance, right? How do you create balance and manage balance?
That's a very good question. I think how do you balance sort of speed and technical execution with compliance and risk over there? I think one thing that I've done throughout my career is basically prioritize or put a lot of emphasis on what I call risk-based design. So when you're thinking about your product development or that entire roadmap, make sure that you're embedding that roadmap with all the regulatory requirements, whether it's the FDA design control requirements or it's ISO 13485. Make sure that those compliance requirements are part of your product development journey. Because if you do that, then you'll find that as you're developing your product, you're going to pick the design features that will completely de-risk the impact to patients. So having that sort of, that regulatory requirements mindset is a very, very important component over there. The other thing you can do is, find the right people who have a good understanding of regulatory requirements, bring the right consultants on board, but make sure you don't lose sight of that because patient safety is the most important thing at the end of the day.
Is this balance like a game of chess where you're always looking 3 steps ahead or 9 steps ahead?
You have to in a way. And the good thing is that there are a bunch of tools you can use. So in our medical device domain, there's a bunch of tools called FMEAs, which are failure mode effective analysis. And you can keep doing this analysis to figure out, okay, what product features do I put in the product so that the risk is minimal? And if there are certain features that increase the risk, well, then they go out, right? So you do that filtering by constantly applying risk management to your product design. I think having that risk-based view is the best way to move fast without compromising compliance or patient safety.
Understood. Suraj, let's switch gears a little bit and start to talk about AI. How are you using AI today?
Yeah, I think a couple of areas. We are using AI sort of at the front end of our product development pipeline. So we are using AI extensively for biomarker discovery. So if I'm developing a new diagnostic test, we typically measure what's called biomarkers in your blood or in your stool sample or whatever, right? So how do I find the best biomarkers? I think that's where AI is being a very, very powerful tool because you can look at very, very large data sets from healthy people, from controls, from cases, and you can use these AI tools to find patterns to figure out what are the best biomarkers that we should be looking at. So it helps a lot with looking at very, very large data sets, helps with pattern recognition, reduces the bias. It enables biomarker discovery at a very, very fast level. So we're using it a lot in that front end. In our current medical device that we're developing for colon cancer, we also have a machine learning enabled algorithm that sits at the back end. So when we run the full test, like I said, we finally basically report whether a sample is positive or negative for cancer. That sort of classification is done by a machine learning trained model. So we're using it at the front end for biomarker discovery and we're using it at the back end for sample classification.
And what about your day-to-day work? Are you big into using AI tools for efficiency?
Yeah. I mean, I think everyone's on ChatGPT now to get instant sources of information. We're doing a lot of sort of, again, machine learning based models for other diagnostic tests. So it's slowly becoming a very integral part of the diagnostic journey that most companies are working on.
Are there certain problems in your field that, that map well to AI, and are there other problems that don't map well to AI?
I think one area where AI will help us a lot, and it's already helping us a lot, is what's being called multi-omics. So today with AI, what we can do is we can basically integrate data sets from many, many different omics. So we have genomics data, we have proteomics data, we have what's called immunomics or immunology data, immune biomarkers data, we have the microbiome. What AI can do is it can take all these very different technology sets and the data from all of them, integrate all of them together. And again, if you have enough number of healthy controls and cases, cancer or whatever, it can take this very, very large data set or data dump and then find out the best biomarkers for you. I think that's one area where AI will just drive the future of diagnostics, is the ability to integrate data from different omics, different technology platforms, and then find the most robust and clinically actionable biomarkers. So if you think about cancer, right? Cancer is such a complex problem that no one technology will be enough. You can't solve all the cancer diagnostics problem with genomics alone. We're finding that we need genomics. We need to look at proteins. We need to look at the microbiome. So all these technology platforms have to work together. And I think that integration is where AI will drive the future of diagnostics.
What else does AI enable today?
I think AI is enabling many other things. I think if you look away from diagnostics in clinical trials, it can be a huge tool in how do I find the right people to enroll in clinical trials? Because what AI can do is it can look at all the metadata that we have for patients. So it can look at all of your EMR data, it can look at your, say, smoking history, it can look at other health conditions that you have, and then tell you whether is this person the right person for enrollment in this clinical trial or not. I think that ability to look at very large data in a completely unbiased way and then make some very nonlinear correlations is what AI is going to really drive.
Let's delve a little bit deeper into the example that you gave for clinical trial selection, right? So what is the main goal of a clinical trial?
The main goal is to basically enroll people who, let's say you have a drug that you're developing for, I'll just give you an example, like an immuno-oncology drug. So you have a drug that's going to target a very particular protein or a very particular marker that somebody has in their cells. Right now, the only way we figure that out is, well, if that marker is present in that person, then we should enroll that person. If you don't have that marker, I won't enroll you because my drug will not act upon you. You don't have the marker. What we have found over the years in immuno-oncology is that even though you have the marker, you may not be responding to that drug. And that's a big mystery. Why? Why are we not responding? Because the marker is there, the drug is supposed to act on that marker, but still there is no clinical benefit that you're getting from the drug. Well, it turns out biology is very complex, and it's not as simple as drug and marker. There are many other things that are part of your, of your system that might be impacting whether the drug will act or not. So AI is able to look at all of the data, the people who have responded to the drug, the people who have not responded to the drug, and it can find maybe 50 other things that the ones who respond are the ones who maybe don't smoke, maybe people who exercise very regularly, who have a certain diet plan, who eat Mediterranean food or whatever. It can find these nuggets of information that you can then apply at the screening level. Well, I will not take you in the clinical trial, Manish, because you have been smoking for the last 10 years, and I know that people who smoke don't respond to this drug. So even though you have the marker, I'm not going to enroll you in the trial because you have a smoking history. So it really makes clinical trial enrollment very, very smart by applying these other data sets.
Very interesting, Suraj. Suraj, let's talk about the future a little bit. Let's talk about, I think 4 things that we want to talk about. I typically call the segment as predicting the future. So we're going to get your take on what's going to happen in the next 10 to 20 years. And we're going to talk about 4 things: life sciences R&D, therapeutics, diagnostics, and product development. Right? I know that's a lot. I don't think there'll be a better person in the near future that I'll get to talk to who can, make these kind of predictions on these areas. So, we're going to spin the wheel and see what you say. And then maybe 20 years later, we'll connect again. Suraj, what is the future of deep life sciences R&D?
I think I'll connect the future of life sciences R&D with the therapeutics and diagnostics as well, because I think they're kind of interconnected. I think we are moving into an era, Manish, where the way we look at problems and the way we think about finding solutions will be very, very different. Because so far, I'll give you one example, right? If you think about therapeutics, because that's one area you asked about, we have hundreds of pharmaceutical companies in the US and in the world who are spending millions, if not billions of dollars, because they know that this particular protein is the protein I want to target basically cure diabetes. I'm just giving you an example. And to do that, they have to understand the protein structure, they have to understand the protein function, they have to understand how the protein is folded, because then they can find a small molecule or they can find an inhibitor that will bind the protein, block its function, and then cure the disease that we want to address. Without artificial intelligence or without this next era that we're moving into, it's a very iterative process where you're trying to make guesses about, okay, this protein structure is like this. If I add this small molecule, maybe it will work, maybe it will not work. And then you have thousands and thousands of small molecules that you have to test one by one, one by one. Is it binding? Is it blocking? Is it binding? Is it blocking? Well, AI can do all of that by simulation in a matter of a few hours. So think about how much time and money and effort pharmaceutical companies have been spending over the last 30, 40, 50 years in this iterative cycle of testing, one molecule at a time. Does it work? Does it not work? Second, third, fourth. All of that can now be done by simulations. You run a simulation through AI. AI tells you these are the 5 molecules that look most promising. Now, as a scientist, I will just go and test those 5 to confirm whether AI told me the right thing or the wrong thing. I don't have to screen 1,000 molecules one by one. I will only screen the 5 that AI told me make most sense. Think about what AI will do with therapeutics with that very simple example that I've given to you. On the diagnostic side, I already touched upon this. The more you can integrate data from different technology platforms, from genomics, from proteomics, from microbiome, from your immune system, from many, many different aspects of your human body, think about the diagnostics we will actually start developing. Think about how quickly we can start diagnosing cancer, even before you have the symptoms. Think about what that will do to the future of healthcare. That is what is now in sight if we make the right moves over the next 10 to 15 years.
And what about product development?
Product development will follow. Product development is a process. It is not a technology. It's a process. How do I incorporate these technology elements to now develop my product in the fastest possible manner? It's a process. It's not a technology, right? So product development will change. More and more AI will start getting into it. There'll be more questions about compliance. What we still need to figure out is what kind of regulatory framework do we have to build around AI? How do we control that? How do we tell the FDA that I figured these 5 molecules make sense because I used AI for it? Will the FDA buy that? Those are things that we have to now figure out. How do we come up with a regulatory framework? How do we think about compliance? Ethics is a big one. How do we think about ethics and AI? Those are all open questions that we have to tackle in the next few years. And product development is a process that will just follow.
Suraj, some people say that if we can live for the next 30 or 40 years, we might live for the next 150.
It's possible.
Is it possible?
I think we're entering an age where you will have so much data about your health at your fingertips that is unbelievable. I mean, we're already seeing it, right? Even today, you have so much more access to data that we didn't have even 20, 25 years ago. You can have all kinds of biomarker data available to you. But I think we are now moving into a space where you won't just have sort of snapshots of your data, that I have my blood work data, I have my PET scan data, I have my colon cancer screening data, whatever. You will have access to sort of integrated data sets. What does this mean for you in terms of risk of developing cancer? What does this mean for you in terms of risk of developing Alzheimer's or Parkinson's disease? What is your risk for developing arthritis in the next 10 years? I think all those things will start coming. The question is, how do you deal with that? Will people then start getting more paranoid and just, keep running to doctors? Will we need a lot more mental health counselors? Or how do you control a world where there is so much data that impacts your health and your well-being?
Suraj, you're going on the road and suddenly you see another person coming from that side and it actually looks like you, and you soon discover it's Suraj of the year 2000. What would you advise him?
Fasten your seatbelts. I think the Suraj of 2000 was a very naive, if I can say that. Someone who came here with just, I want to learn science, I want to solve one problem at a time, I want to be very good in genetics or cell biology or biochemistry. We're living in a very, very turbocharged world today. That one focus will not work. You have to understand every aspect, every technology platform that's out there. You've got to pick the best ones, put them together, integrate, and then find the best one that works for you. I go back to what I said, solutions focused, technology agnostic.
Awesome. Suraj, this has been a fascinating conversation. What else would you like to share with our audience?
I will just say, if there's anyone out there who's considering a career in life sciences, this is the golden time. I think some of the best discoveries in this area are going to be made in the next 15, 20 years. I would say if people are looking or thinking about career options, a great time to be someone who can bridge a very good understanding of biology with computational science. I think that marriage of computer science and programming and data science and bioinformatics with deep understanding of biology and cell biology and disease and all of that is a very, very fascinating area to consider as a career option.
Awesome. You seem to be still fascinated with the basics, Suraj.
You never lose the basics because at the end of the day, I can think of any technology platform, but it is that small thing that's gone wrong in your cell which is causing the disease. So to understand that is still basic science that is required.
Understood.
These are all just turbochargers that sit on top of it.
Suraj, thank you so much. This has been a fascinating conversation. Today we were joined by Dr. Suraj Saksena. Dr. Saksena is the Vice President of Product Development at Universal Diagnostics. Suraj, thank you so much for being on the Inference Show.
Thank you, Manish. Enjoyed it.